thanks for the introduction. Thanks for the invitation to be here. So as Becky mentioned, I'm Shilpa Kobren, and I'm going to be talking about a paper that we published in Genetics and Medicine earlier this year called Commonalities Across Computational Workflows for Uncovering Explanatory Variants in Undiagnosed Cases. So probably as many people in the audience may know, genetic disorders that folks are born with are estimated to cumulatively affect almost 1 in 15 people worldwide. But if you take a look at the prevalence of any single one of these genetic disorders, what we see is that the vast majority of them are extremely rare, right? We see this long tail effect. And sometimes they're so rare, they're affecting only a single individual or they're only found in a single family. And what that means is we can't use traditional case control analyses to figure out which genetic variants are giving rise to the disease. So just to refresh your memory, in a case control setting, you would have some number of cases, more than one, preferably from unrelated individuals, and you can construct a well-matched control set. And when you look at the genomic sequences for those in the case group versus the control group, if you're lucky, you'll find genetic variants that are unique to the individuals with the genetic disorder that are absent from the well-matched controls. Now, on the other hand, if you're in this ultra rare disease setting and you only have a single individual who is exhibiting the genetic condition of interest, you can't use your case control analyses, right? Instead, what you do is you have access to their medical records, clinical notes, family history, and also their genomic data. So there's thousands of variants that can be uncovered in a person's exome and genome. And what you have to do is look at various lines of evidence, such as phenotypic associations and existing disease associations for genes of interest, the expression of those genes, scores such as selective constraint in humans or predicted pathogenicity, to try and whittle down this giant list of variants down to the few diagnostic variants that could be giving rise to the genetic disorder. So there are a lot of questions here in this ultra rare disease setting. First, how do you find the variants in the first place from genomic sequencing data? And we know in general that all of these different lines of evidence are considered, but in what order? What's more important or less important than something else? And of course, what methods and tools and data sets are actually used in practice? Because we can talk about in general how selective constraint is used, but which data sets and how? And are there standard practices that are used across medical institutions or clinical genomic sequencing centers? And of course, there are multiple different medical centers that are trying to solve cases like this. There's multiple different disease consortia and universities. And how equivalent are their different workflows? And kind of in the same vein, where are the biggest differences between workflows and what do those differences mean? So these are questions I had about the diagnostic process in this ultra rare setting. And to start to answer these questions, I turned to the Undiagnosed Diseases Network. So the UDN is NIH funded, and it's the largest undiagnosed initiative in the United States. Currently, there are 12 clinical sites that are part of the network. One of them you'll see in the top right is Harvard Medical School and 3 Harvard-affiliated hospitals, which includes, of course, Boston Children's. And in addition to the 12 clinical sites where patients are evaluated and these cases are solved, there is a DNA sequencing core at Baylor Genetics in Texas. We have 2 model organism screening centers and also a metabolomics core. So how does one enter the UDN? Usually what happens is you have a person with a particular ailment and they can spend years on this diagnostic odyssey, basically hopping from specialist to specialist between different medical centers until finally a physician will refer them to the Undiagnosed Diseases Network. At that point, their case is routed to a case review committee, and there's certain questions that are asked. First, clinicians will take a thorough look at the available medical records and the application letter to confirm that there isn't already a diagnosis that's been found that's sitting somewhere in that data. Want to make sure that a lot of the tests that would or should have been done have already been done, and they really didn't reveal a diagnosis. Can a UDN evaluation help? And very importantly, is the patient and hopefully other family members available for genomic sequencing? So if these are all true, a patient is eventually accepted to the UDN. And here are just some numbers from March of this year. It's not a very big data set, but remember, the UDN is selecting for kind of the toughest cases that have stumped clinicians nationwide for years. The primary symptoms are also kind of all over the place. So it's a data set that's not made up of just one type of condition, although neurological and usually neurodevelopmental is kind of the biggest group, which tells us a little bit about what we know and what we don't know in medicine. So here, our diagnosis rate is just about a quarter. And of course, that's not very high, but given the cases that are accepted to the UDN, it's actually pretty high, but there's definitely room for improvement. So once a patient is accepted, the first thing that happens is a clinical evaluation. And this happens at one of the clinical sites in person. And usually family history and symptoms are collected. And ahead of time, the case review committee would have looked at the patient's medical record, and they would use this clinical evaluation to set up meetings with relevant specialists. And there's also further labs and tests that can be performed. The clinical evaluation can take up to a week, and the patient plus family members are usually flown or travel is reimbursed to the nearest clinical site in the US. And also importantly, genetic sequencing happens. So here I've colored in red the person who has the genetic condition, and in gray folks in the family who do not. So DNA sequencing of affected and unaffected family members happen. As you recall, we have a DNA sequencing core, which does the sequencing. And in addition to getting all of the raw data back, clinical sites also get access to a clinical report. And I wanted to show you exactly what appears on that clinical report in accordance with ACMG guidelines. So first, is there a variant in a disease-causing gene directly related to the clinical phenotype? That's kind of your answer. The second is looking at pathogenic variants in disease-causing genes that aren't yet known to be related to the clinical phenotype. And then finally, carrier status for certain recessive Mendelian conditions. Only in about 10% of cases does the clinical report list any variants at all, right? These are really hard cases. Usually what's gone wrong is in a gene that we don't yet know to be disease-causing. So then we have a manual case review where clinicians and genetic counselors will look at results, and this happens simultaneously with computational variant prioritization. And this is where there's an analysis of the genomic sequencing data to try and find candidate explanatory variants. Once one or a few candidate variants are found that could be explaining this person's condition, we look for matches in other human beings in databases such as Matchmaker Exchange. We look for matches in other species, or we develop new experimental models, in vivo or animal models. And finally, which isn't listed on this slide, we also look for secondary phenotypes. So supposing that we do have a variant match in another human or another animal, we would look at phenotypes there and see if they exist in our original patient as phenotypes that we may have originally missed. So for the rest of my talk, I'm going to focus just on this piece, the computational variant prioritization. What's actually happening here? What do we do with the sequencing data to try and find candidate explanatory variants? And so we met with bioinformaticians and genetic counselors across 11 of the 12 clinical sites and the sequencing core to figure out what are the steps that go into this process. And just as a little bit of motivation, the reason you would want to do this are related to the questions I had asked a little bit earlier, like what are the commonalities? But getting a sense of what different sites are doing also gives you a good idea for reanalysis. If there's certain steps that have worked for other clinical sites that we haven't tried yet, and vice versa. And also for sites that aren't the UDN, who are interested in seeing what types of analyses are going on. So all the steps I was able to group into these kind of broad categories. First, alignment. The raw sequencing reads are aligned to a reference genome. Then SNPs and indels are called. And here's actually a big piece that I'll talk about. Structural variants are also called once reads are aligned to the reference genome. Once you have your variants, there's a giant process of annotating those variants with whether they fall into known disease genes, how the variants segregate in healthy human populations, predicted pathogenicity, predicted functionality, and inheritance. Variant annotation is tightly coupled with variant prioritization and variant filtering. So in addition to looking at these annotations to try and whittle down your set of variants that might be causative of the disease, folks also have started to integrate transcriptomic sequencing, such as RNA-seq. There are structured phenotypes, which I'm also going to get to. And of course, to manage this whole process, there are a variety of different workflow management tools and collaboration tools. If we look across all the 11 sites plus the sequencing core, for what tools they're using to carry out these different tasks, what you'll see is that there's actually a large diversity of approaches that are used to try and solve the country's most difficult diagnostic dilemmas. And quality control is something that we should never skimp on, and I'll go into a little bit of detail there too. So before continuing, I really wanted to emphasize that a research pipeline, which are the types of pipelines that I'll be presenting, are not the same as clinical pipelines. There are standards that exist for clinical pipelines, like CAP accreditation and CLIA certification. Research pipelines can get away with being a little bit more wishy-washy, and they also change often. And again, because they're not clinical pipelines, they aren't standardized. And the other big point I wanted to make is that standard practice is not necessarily the same as best practice. So there's a lot of tools that are used across many sites that you would consider to be consensus practice or standard practice. Often it corresponds to the best practice, but in this space, there are so many new tools and methods being developed all the time that it can take a little bit of time for certain methods to organically bubble to the top and become standard practice. Okay, so first I'm going to quickly talk about the alignment step. Alignment is the process of going from FASTQs, unaligned reads, to BAMs, which are aligned reads. And actually, functional equivalence standards have been established. So this is figure 1 from the Nature Communications paper, I think put out from the Centers for Common Disease Genomics in 2018. And BWA-MEM is one of the 2 programs used for alignment, Illumina's Dragon being the other. And I just wanted to add a note that this was true at the time of writing, but at the moment, the Broad is working with Illumina to standardize their alignment pipelines, and graph alignment is also being explored in that collaboration. Quality control, super important, like I mentioned. And I'm going to breeze through this slide just because the sequencing laboratories where you would get your sequencing data already perform this basic quality control in FASTQs and BAMs. So sites have to worry about this when you get research genomes instead of clinical genomes, or if patients come in with their own sequencing data, you would want to redo some quality control. So kind of basic things at the FASTQ level, your number of reads between your 2 paired files should be the same. The quality of the reads should be pretty good. The 2 tools that are used across the board are FastQC and FastP. Now for BAMs, there are many more tools that are used, but they're all kind of getting at the same thing, which is looking at your read depth across the entire genome, the exome, how many of your reads actually aligned well in proper pairs, and how much of the genome was covered. Okay. So once reads have been aligned into BAMs, the next major step is actually calling SNPs and indels from those aligned reads. So the majority of sites are using GATK's best practices workflow. So a lot of folks who work in bioinformatics would be intimately familiar with this workflow to call SNPs and indels. There are a few other callers. So Google's deep variant is used in addition to GATK by one site. And there's a polybase implementation available in real-time genomics that's also used in addition to GATK by another site. The sequencing core uses Illumina's Dragon, and 3 other clinical sites also use Dragon. What's important here actually is this thing called joint calling. So you can imagine if you have a bunch of aligned reads, you can look for variants from the aligned reads in a single individual. But what happens in practice across every single one of these sites and the sequencing core is variants are called across samples. And the reason for that is to rescue low coverage true variants, to accurately model false variants, and this is very important, especially within rare disease, to provide homozygous reference genotype calls for any variant found in the cohort. What that means, right, is if you have a kid who has a particular variant, you want to maybe know that that variant is really not present in mom and it's not present in dad, right? It's a potential de novo event. And you would need a homozygous reference call in both parents in order to come to that conclusion. So in practice, joint calling, you can jointly call members of the same family together. At 2 clinical sites, individuals are their variants are jointly called with other UDN patients from the same site. And sometimes healthy patients, internal or external to an institution, are all jointly called together. So the NIH is doing this. I believe Brigham Genomic Medicine does this. UCLA does this. And in Columbia, they also do that. Once you have your SNPs and indels, quality control, again, super important. And so quality control at this level can be split into kind of 3 categories. First, your sequencing quality has to be acceptable. Your sequence samples have to match your expectation. And there's actually a number of horror stories across the sites of the UDN where if you skimp on this particular step, you can miss that samples have been mixed up and so forth. And also a low rate of Mendelian violations. So first, this one's pretty straightforward. Is the sequencing quality acceptable? What I'm showing you here is a chart. The vertically labeled columns refer to tools that are used to perform these different tasks that are listed in the rows, and in the cells are the number of different sites that are using that particular tool to carry out that particular step. So these ones are pretty standard, checking your transition transversion ratio, checking your het to homozygous site ratio, making sure that you have mostly SNPs and some indels. And then, of course, looking at your distribution of alternate read depth, your variant quality and genotype quality to make sure you don't have any outliers. This is the sequence samples match expectation. This is where, like, if you miss this step, things can go very wrong. So ancestry, for instance, should match your expectation. Usually when ancestry really doesn't match, that's a good indication of a sample mix-up. So you can imagine up until this point, you've got perfectly good reads. They aligned perfectly well. The variants look good. They just correspond to the wrong person. And it is disastrous to continue with your analysis without having discovered that at this step. And also relatedness between individuals. This basically is where you can have paternity checks. If a family member isn't as related as you thought, sometimes relatedness is very high and 2 parents may have had a higher blood relationship than they thought. And of course, if there's perfect relatedness, you've got a sample duplication. And finally, Mendelian violations are another thing that you have to check for in the rare disease space. Sometimes Mendelian violations are important. An example is a de novo variant, for instance. But you want to make sure that your family structure looks good and the variants that you're detecting looks good. So here are just some examples of the types of things that are checked for. I'll get into it a little bit more later, but de novo variants are actually plagued by false positives. If you just look for variants present in a kid with a disorder that are absent from both parents, you can get in the thousands of variants across the genome. And we know that there are actually far fewer. And so, for instance, you can look at something like de novo DB, which will tell you frequencies of de novos, and a really high frequency means it's probably not a real de novo. And novocaller is used here at BGM, salvage pathway used at the NIH, but these just call de novos directly from sequencing reads, so they take into account depth, read depth in both parents in order to call true de novos. Okay, so the next major step, and this is actually something that is very new. It makes sense that UDN sites are exploring it, but a lot of medical centers don't yet do complex variant calling. So first, what are structural variants, right? These refer to large insertions and deletions in the genome, also duplications or other expansions of certain regions of your genome, which are called copy number variants. You can have short tandem repeats, so like a little short section, nucleotide section that's repeated over and over again, that can count as a structural variant. Translocations, where you've got an entire section of your genome that's hopped to a different part of the genome. And then inversions, where DNA can be detached and then reattached in the opposite orientation. So structural variants are actually really difficult to detect. And the reason for that is when the genome is sequenced, it's actually broken into a bunch of short reads that are then assembled onto the genome. And these little short reads that are sometimes only 50 base pairs long, they rarely span the length of a structural variant. And so computationally, it's really hard to find these types of structural aberrations. And as you can imagine from what I described here as potential structural variants, you actually would search for them computationally using very different approaches. And so different SV calling tools will only identify a subset of possible structural variants. And so in practice, multiple different callers have to be combined and considered together. So here's just a table showing you the list of different tools that sites within the UDN are using to find structural variants from your sequencing reads. And I order them based on prevalence across sites. So the most common tool, MANTA, is it's actually run by the sequencing core, and those results are provided to the clinical sites, which is why all clinical sites are considering MANTA results. Expansion Hunter, which appears right below it, is used to detect short tandem repeats, and I know that people really like this tool. It's being used across like the UK 10K project and the 100,000 genomes project and so forth. The two specific functions from GATK, they're called by wrapper tool, XHMM. And remember I said that in practice, you have to be calling multiple different SV callers to get a more comprehensive set of structural variants, which is why wrapper tools are so prevalent when actually finding structural variants. For instance, here's another one. Parliament 2 is being used, I think, more and more often, and it calls a bunch of other methods. And again, you can probably tell from this table that there's quite a bit of diversity here in terms of which tools are being used by which groups. Quality control again. The big question for structural variants is, are these variants real? And the second is, are they rare? So this was actually surprising to me when I first started learning about structural variants, but it turns out there are common structural variants. There are structural variants that happen in perfectly healthy people that are not at all related to disease. And so when you uncover a structural variant in someone with a genetic condition, you want to know if it's pathogenic and not just, is it real? So in order to answer these questions, multiple different databases have to be checked because, I mean, we already know that the UDN sites are all using different SV detection tools, but different databases are also using different detection tools. And what that means is if you look in one particular data set for your structural variant of interest and it's absent, it could mean that it's actually really rare in the population, or simply that the method you use to uncover that structural variant was never run on the data in the data set that you're looking at. And so the two tools, well, Anode SV as a tool and Nomad SV as a data set are used most frequently for quality control of structural variants. Okay, so now that we have our variants, we've got SNPs, indels, presumably some structural variants as well. The next huge piece of the pipeline, and this is really where we start to see a lot of diversity in what in what is going on across the sites is variant annotation. So I'm just going to start with the first item here, which is looking at variants that fall into known disease-associated genes. ClinVar and OMIM, possibly no surprise, are used by every single one of the UDN clinical sites and the sequencing core. And just to remind you about what is in the clinical report, right? I underlined it this time. We're looking at known disease-causing genes, right? This is the first thing that people look at, whether there are any variants that are falling into known disease-causing genes. In addition to ClinVar and OMIM, there are a few other data sets that people are also looking at to see if there are variants that have been associated with disease that are uncovered in your patient. So after looking at variants falling into known disease-associated genes, the next big ticket item that everyone is looking at is variant segregation in healthy human populations. So what does that mean? Right, so for folks attending this seminar, population in general, we've got a lot of healthy people. And it turns out that when you look at the genome sequences for these healthy people, there are these variations between people that are presumably unrelated to disease. And so we can actually use this information to figure out if certain variants we're looking at in a person with an undiagnosed genetic condition, if those variants are causing a disease or not. The idea is a really common variant that's found in half of all healthy people is not disease relevant. So these are the three biggest databases that are used in the UDN. Presumably they're used by other medical centers that are also trying to diagnose patients. How are these data sets used? Well, most sites are actually just implementing a minor allele frequency cutoff. So basically any variant that's found in more than 1 in 100 people or 1 in 1,000 people, they're kind of discarded as likely not responsible for the disease in question. In dominant cases, this minor allele frequency is pushed even lower, and sometimes in recessive cases, it can be raised to try and rescue more variants to consider. 2 sites eliminate variants found as homozygotes in 3 or more individuals. And at the NIH site, instead of using the minor allele frequencies that are provided in these data sets, they actually compute a 95% Wilson confidence interval and in order to retain rare variants. And just like to give you an intuition of what that means, supposing you had one spot in the genome where you were only able to get good coverage and good sequencing for 10 people, and one of them happened to have a variant, right? That translates to 10% for minor allele frequency, but you don't really know that, right? Like had you actually had coverage across 100,000 people, that one person could have been the only one. And so that's what the Wilson confidence interval can help you with. 5 sites are also using local data to flag variants that they found in other individuals at their institutions. Of course, because at your institution, you also have medical records for a lot of these patients. If you've got a patient who is asymptomatic or differently symptomatic, probably that variant is not relevant. And here are just some other data sets that are also being used. I wanted to mention TopMed is used specifically when the inferred ancestry is non-European or when the NOMAD minor allele frequency is 0. And this is important because there are some sites in NOMAD that have a minor allele frequency of 0, and it turns out they're not actually that rare. There is just low coverage in that region, and NOMAD refused to report on variants. So looking at TopMed, especially in HG38, especially in non-coding regions, is super important. And then there are some kind of population-specific data sets that are used when inferred ancestry matches what that data set is used for. So in addition to looking at the actual frequency of that specific variant, we can also use these what are called selective constraint scores. And so all of these SNPs and indels that we're seeing across healthy humans are not uniformly distributed, and they don't all have the same impact, of course, right? It turns out that there are patterns to how that genetic variation is laid out across healthy humans. And we can use that information, the distribution of variants and what impact that variation has, to capture how certain regions of the genome might be intolerant for loss of function variants, right? So if you had a particular gene that had quite a bit of variation in healthy humans, except all of that variation was synonymous, that's good evidence for that region being intolerant for a loss of function variant. So here are the different selective constraint scores that are used in practice. I just wanted to point out here that using selective constraint scores is not necessarily part of clinical pipelines yet, but so you can see like the Baylor-Seq column doesn't use any of these, Baylor-Seq being the sequencing core that provides clinical sequencing. But the other UDN sites are all using some form of selective constraint score. So this is an idea, for instance, of the types of scores that would be useful in this research setting. Okay. So once you've looked at variants that are falling in known disease genes, you've kind of filtered out to the really rare variants or regions that are intolerant for loss of function variants. The next thing folks look at are predicted pathogenicity of variants and the predicted functional consequences of variants. So variants of unknown significant, VUS, are the most abundant type of variants that are uncovered. And so you have to kind of use these predictive methods to determine if that variant might be pathogenic or not. So here's just a table showing you what types of tools are used. Polyphen2 and SIFT, they're the oldest, they're the classic ones. Most people are using them. But if you look under this second section here, the ensemble pathogenicity predictors, it turns out CAD and REBEL are the most commonly used tools when looking at predicted pathogenicity. And so I just mentioned the cutoffs here. Usually folks consider a CAD-FRED score greater than 25 to be a variant of interest, or a REBEL score greater than 0.3. And I'm gonna go back really quickly just to say that most of these predictors only work for coding regions. And that's really important because we now have the ability to do whole genome sequencing. And so not only are the predictions bad in the non-coding space, but even if you found a variant that you think might be really interesting in the non-coding space, it is notoriously difficult to experimentally validate those. And a notable exception are non-coding variants that could affect splicing or expression, because those you can find experimental evidence showing what the impact of that variant is. And so here are just some of the tools that are used for predicting the splicing impact of a variant or the expression altering impact of a variant. Splice AI is the most commonly used tool for this. Okay. So up until this point, right, all of these different annotations worked if you only had one individual with your disease, and that's the only person you're looking at. But inheritance is a really important factor, and you can get at the inheritance of different variants if you have sequencing data for parents or other family members. So what does a sporadic case look like? When I say sporadic, what I mean is you have a kid who's affected and it seems like nobody else in the family is. 2 unaffected parents, other unaffected kids. A suspected recessive case, you can have a sibling who's also affected, again, 2 unaffected parents, or you can have kind of a larger family tree where some disorder pops up through generations. But again, the important piece is that you have 2 unaffected parents and an affected kid. So what types of variants could be causing a sporadic or a recessive condition? So what I'm showing you here, just 2 kind of cartoon images of chromosomes or pieces of the genome, and the orange ovals I use to represent variants. So of course, mom has 2 sets of chromosomes, dad has 2 sets of chromosomes, and a kid will inherit 1 chromosome from mom and 1 chromosome from dad. So when you look at the affected proband, you could have a homozygous recessive variant that is causing the condition. And this means that the kid was unlucky enough to inherit the exact same variant from both parents, which happens to cause a disease. And homozygous recessive variants are actually extremely uncommon, except in cases where parents have a high degree, or they're related. Another type of variant is a de novo variant, a variant that neither parent had that occurred spontaneously in the kid. And that de novo variant happens to be causing a disease. Finding de novo variants, like I mentioned previously, plagued by false positives, because usually what happens is the variant looks like the kid has it and neither parent did, when in fact it was inherited from a parent. It just so happens that the sequencing was of low quality or depth in that parent at that spot. Another possibility is something called uniparental disomy, where instead of inheriting a section of the genome from both parents, you actually, one section came all from the same parent. And somewhat similar is inheriting a large deletion. And so both of these cases are detected as a stretch of homozygosity in a kid. And what this is an example of, or this is a case where you can have a recessive condition in the kid, even though only one parent was a carrier of the disease. And finally, there could be compound heterozygous variants, right? So these are two different variants that together are going to cause the disorder, maybe because each of them has a slightly deleterious effect. And when you have two kind of non-functioning copies of the same gene, you're going to get it. Going to get the disorder. So this is the most common cause of recessive conditions, but as you can imagine, there are so many combinations of variants to consider in the compound heterozygous case that in practice, what has to be done is what's called pre-filtering. So you're only going to consider extremely rare variants with a high predicted pathogenicity in order to look at pairs of potential compound hets. Okay, so these are all the different ways we can annotate variants, but there are tools to actually do this annotation, right? And so here's just a list of them. Ensemble's variant effect predictor, or VEP, is the most common tool used to assign variant annotations to a VCF file, basically. And I just wanted to point out that some sites don't explicitly use variant annotation tools. They run these automated pipelines where annotation is built in. The different tools, in addition to kind of tacking on certain annotations, the functional consequences assigned are also important. And I think that's one of the reasons that people tend to use VEP, because their functional consequences for what the variant is going to do, for instance, is it a missense or a frame shift and so forth, are some of the most accurate. Okay, so as I mentioned earlier, variant annotation is very tightly coupled with the entire variant filtering and prioritization. So when I was mentioning the different types of annotations, I was simultaneously saying what some of the cutoffs are for variants to be considered. So that's the filtering process. During this prioritization process, there is an opportunity to integrate other types of data. So you can integrate RNA-seq data and also structured phenotyping data. So first, I'll just talk a little bit about transcriptome sequencing, right? So RNA-seq is getting the sequences of the transcripts present in a person. And as you can imagine, RNA-seq is going to be very dependent on the tissue you have access to. So you can use it in this DNA first approach, right? So supposing you analyze the genomic sequencing, you found some variants that could be affecting expression, could be affecting splicing, then you would get RNA-seq from the relevant tissue and confirm whether expression was indeed affected or whether splicing was indeed affected. As you can imagine, this is mostly going to be relevant for blood phenotypes or muscle phenotypes. But in the UDN, there are some patients who unfortunately passed away who had neurological disorders, and we do have RNA-seq brain samples from those patients. On the flip side, you could go RNA first, right? Supposing your genomic sequencing didn't reveal any variants of interest, you can then do RNA-seq and sometimes immediately see genes that are very aberrantly expressed relative to healthy tissue match samples, for instance, available in GTEC. So here is a table showing you tools that are used, what the purpose of that tool is, and which sites are using them. And you'll notice I only list 5 sites here. Transcriptomic data is one of those things that a lot of different sites want to be using, and they're ramping up their RNA-seq analysis pipelines. But at the moment, it's still very new. And so not every site yet is ordering RNA-seq and analyzing RNA-seq. So the first set of things you do, pre-process the reads, align them, and then kind of map them to exon and gene and figure out how much transcript you have per exon and per gene. DESeq2 and EDGER are 2 really common programs used for analyzing differential expression. And then, of course, as I mentioned, detecting expression anomalies, detecting splice junctions, detecting splice junction anomalies. There are several different tools here that can be used. And again, this is an area where there's a lot of diversity in tools, which suggests that this is a very exploratory analysis still. There are no standard best practices yet for analyzing transcriptomic data. And a lot of new methods continue to be developed and used. So next, integration of structured phenotyping is another way that, or another thing that can help with variant prioritization. So just wanted to mention that, of course, deep phenotyping of patients is critical to the overall UDN process. If you remember, looking at medical records, the in-person clinical evaluation, these are super important in general for diagnosing individuals. So when I'm talking about the computational piece or the computational pipeline, it turns out that in the UDN and actually across other consortia as well, symptom terms are standardized via the human phenotype ontology. So it's not just free text. And HPO terms are related to each other in a tree-like structure. And so computational tools can actually reason over HPO terms in order to generate gene panels, right? So genes of interest related to a phenotype that can complement manual efforts. And there's lots of tools that can be used for this particular process. So first, here are a set of tools used across the different UDN clinical sites that take in a set of HPO terms for patients, so phenotype, as well as either a list of genes or directly a VCF, and it will do variant prioritization for you based on phenotype. So this is cool. One caveat is this is entirely dependent on which variants you feed in. It could be that your variant caller missed a few variants, or you gave it genes and those genes did not actually contain the variant of interest. These tools are going to be limited by the genotype input. There are also a set of tools that just do gene prioritization based on phenotype input. So given just the set of HPO terms, which genes might be relevant? And this is really nice, especially when you want to focus on a set of genes. For instance, you suspect that a variant of interest might be in the non-coding space and may have been missed by some of the other methods that focus just on coding mutations. And then finally, there's some databases that are used pretty often that contain gene to phenotype relationships. So the Monarch Initiative, I think, is one of the biggest browsers that contains this information. Mouse genome informatics is used either in the computational pipeline, but it's also heavily used by clinicians and genetic counselors who analyze variants manually after the fact. And the HPO website also contains HPO gene annotations. So I went through all of these different steps, right? We start with alignment, variant calling, annotating the variants, attempting to filter down the variants based on all these different factors, integrating other types of data. So this is a gargantuan project, right, for every single site that does this. And we need workflow management tools and collaboration tools, because bioinformaticians are involved, genetic counselors are involved, clinicians are involved, and they all need to be looking kind of at the same set of variants or genes, taking notes, sharing notes with each other. And so workflow management and collaboration tools are very important for any of these computational pipelines trying to find explanatory variants. So here are the top used tools, current as of fall 2020. I wanted to make that disclaimer because a lot of these tools are changing. There also seems to be kind of an explosion in the startup scene with different companies that advertise themselves as being able to automatically analyze sequencing data with phenotype information to try and prioritize variants. But no surprise, maybe, Excel spreadsheets are the top tool. It's the top tool used for sharing variant level information, sharing notes about variants. Box and Dropbox is used to store variant information, patient information, notes, presentations, and so forth. So the main takeaways I want to leave you with are, first, research analysis pipelines are not limited to existing disease knowledge the way that clinical pipelines can be, right? So again, clinical pipelines are looking at known disease genes, but the research analysis pipelines that are implemented at UDN clinical sites can discover novel genes. The pieces of the pipeline that differ between sites, right, these divergent aspects, they first reflect promising avenues for case reanalysis. What UDN pipelines do, the tools that are used, the data sets that are used, the order in which things are looked at, it's not random, right? These things all happen because they help solve cases at that site. And so if a site is doing something that another site isn't, it could be that that is something that would be interesting to try, especially on the remaining still undiagnosed cases. And of course, places that seem to differ the most across pipelines really reflect where technical improvements or theoretical improvements would be impactful. It's really cutting edge area. And there hasn't been kind of community established standard practice there yet. So some examples are looking for experimentally verifiable predictions for non-coding variants, automatically integrating additional biomedical data such as RNA-seq, and improving SV detection, specifically the specificity of structural variant detection. And of course, the tools that are used across sites, right, the standard practice tools, what we found is they have particular characteristics. First, they have been convincingly evaluated by independent parties, right? They are easily incorporated into existing workflows. So this is important. Tools that operate at the VCF level, at the BAM level, and so forth, that don't require a lot of customization are going to be used more often. And of course, having a maintained implementation. And a lot of folks have complained about having found a really great tool that unfortunately was written by some graduate student who moved on. And the lack of maintenance for a lot of really great tools is a problem. And so with that, I'd like to acknowledge the UDN Tool Building Coalition Working Group. These are the folks that this is the group that I run and the folks that I work closely with in the UDN. Zach Kohane, who is my direct mentor, who really encouraged collecting all this information and writing it up. And then the folks who provided details about their pipelines and put up with all of my questions and meetings across sites. And so with that, I'd like to say thank you. And I'll take any questions.
All right, thank you so much. So do you have any question for Shilpa? If you have a question, please unmute yourself or type your question in the chat window. Maybe I can ask one question, so give other people some few minutes to think about the question. I think that you talk about the SV detection. So I was just wondering, in the UDN database right now, you should have the whole exome sequence data, whole genome sequence data. So do you think about, do you also have non-read sequence data to detect this kind of structural variation?
Excellent question. So a lot of folks are looking into using long-read sequencing data. Long-read sequencing data isn't currently covered as part of the UDN. And so the sequencing core at Baylor Genetics isn't providing long-read sequencing data. But the site at, I believe, Washington University and UCLA, they have paid for external sequencing of UDN patients using long-read sequencing data. one of the big holdups is having a convincing method that works to find those structural variants. So that, like the folks at the UDN sites, they aren't just applying tools. I failed to mention that a lot of the tools listed that are used across sites, that are state-of-the-art, have been developed by bioinformaticians at these different sites. And so they are developing tools to look at long-read sequencing data.
Great. I think we have one question from Kiko. So I think this is a great question. Are there pipeline to analyze ritual transposon insertions?
Oh, that is a good question. I believe there are tools to do that, and I cannot think of any off the top of my head.
Yeah, there's one tool, it's called TEA.
Yeah, that sounds right. I know that at the Stanford site, Matthew Wheeler is also actively working on tools for looking at mobile element insertions across UDN data too.
Okay. So for the jargons, so we actually also have jargon for our CRDC cohort. So did you hear any feedback for jargon? How good they are?
Yeah, I think people are super happy with Dragon. The one thing that makes Dragon a little bit difficult is, of course, it's not open source, so it's really hard to establish functional equivalence. And that becomes an issue when you're dealing with a consortium like the UDN, or which is becoming more and more common where you're combining data across cohorts. So for instance, right now, we have UDN data, but we also have collaborations with Care for Rare in Canada and collaborations with Children's Mercy Hospital in Kansas City. And unlike BWA-MEM, where there are functional equivalence standards and you can compare what went into processing that data, with Dragon, it's not so easy to do that. And so that's actually part of the reason for this ongoing collaboration between Broad and Illumina right now to go through the functionality of Dragon specifically for alignment to figure out how to make it functionally equivalent between what's open source and what isn't. But Dragon does an amazing job. I think that's why it's used so often by so many different people.
Great. Yeah, it's really great talk. Do we have any other question from the audience? If not, thank you so much, Shilpa. It's really great talk.
Thanks for the invitation.
Thank you. Bye.
Bye.